Hello and welcome to my tutorial on organelles. So I'm going to talk about their structure and function. So first off, we're going to start with the nucleus. So the nucleus has a little bit more detail than you're used to. It has the nuclear envelope with a nucleus, which is the less dense part of the entire nucleus, and nucleolus, which contains which is more dense than the outside nucleus, that contains all the genetic information that is needed. The whole nucleus contains genetic information, but the nucleolus is the most concentrated part of the nucleus in terms of DNA. And the rough endoplasmic reticulum is on the outside. This is used to synthesize proteins when RNA is released outside the nuclear pore. So now we'll just look back at the notes, and it's the largest organelle, so it shows darkened patches known as chromatin. It is surrounded by a nuclear envelope, and it's a structure made of two parts, or nuclear pores, which go right through the envelope. So it holds a large enough for relatively large molecules to pass through, and the nucleolus is the dense structure in the middle. The nucleolus contains all the cell's DNA and proteins. The chromatin consists of DNA, and it has instructions for making proteins. Some proteins regulate the cell's activities. When the cells divide, chromatin condenses into visible chromosomes, which look like this, sort of like a big X, and don't forget the chromatin, it kind of coils itself into these small chromosome things, but they're more dense, so you can see them under a light microscope. And the nucleolus makes RNA and ribosomes. They pass into the cytoplasm, and the proteins are assembled in them. So now we'll look at the endoplasmic reticulum. This is what you saw just earlier, but I've got another picture of one. So there's the nucleus and the nuclear envelope. So once the RNA is released, it synthesizes proteins on the ribosomes, and then the smooth endoplasmic reticulum deals with the synthesis of lipid molecules, or fats. So it consists of a flattened membrane brown sacs called cisternae. They are continuous with the outer nuclear membrane, and the rough endoplasmic reticulum is studded with ribosomes. Smooth endoplasmic reticulum does not have ribosomes. The rough endoplasmic reticulum transports proteins that were made on the attached ribosomes. Some of the proteins may be secreted from the cell, some placed in the cell surface membrane, but smooth endoplasmic reticulum is involved in making the lipids that the cell needs, so I mentioned that earlier. And the Golgi Apparatus are a stack of membrane brown flattened sacs. They receive proteins from the endoplasmic reticulum and they modify them. They repackage them, and they may add sugar molecules to them. They also modify proteins and they package them for secretion from the cell membrane. This is done via the process called exocytosis. So if I look at the Golgi apparatus, there's the cyspace, and the cisternae are all the flattened sacs. Lumen, which is just the narrow part of the sacs, and the vesicles is what they release. These contain the newly formed proteins. So if we go back, mitochondria are spherical, and they have two membranes separated by fluid-filled space. The inner membrane is highly folded to form cristae, and the central part is called the matrix. This makes the energy required for the cell. It makes ATP in animals, and it's also known as the universal energy currency, as it's needed to do pretty much everything in a cell. So there's your cristae, and there's the outer membrane, and the intermembrane space is where it's all made. It's then emitted out of the outer membrane, where around the dense parts is the ATP just being released, being emitted, Chloroplasts are only found in plant cells, and the cells of some protists or bacteria, and they allow a plant to photosynthesize, and they also allow it to make ATP as well inside the thylakoid membrane, but more about how they make ATP in A2 level. For now, you just need to know that they make glucose and water in the granum and the thylakoids, and the stroma is just the aqueous space where H plus ions reside for ATP synthesis, but more on that in A2. Lysosomes are spherical sacs containing the digestive enzymes, and their role is to break down materials from dead or decaying cells. They can also break down microorganisms. So if we look at a picture, this picture is the lysosome, and the phospholipid bilayer is the cell membrane. That's what would be yellow on the outside. It's represent the lipid molecules, and there's an enzyme mixture inside, and they're released in order to ingest pathogens, disease-causing molecules, or just decaying parts of the cell. So definitions now: the term organelle is referring to a piece of a particular structure of a cell that has a specialized function. Some organelles are membrane bound, others aren't. All perform a particular role in the life processes of a cell. So we have a look at the organelles without membranes now. Ribosomes are really tiny organelles, and they're found free in the cytoplasm or attached to the endoplasmic reticulum, and they consist of two subunits. They kind of look like this. They're where protein synthesis occurs, and they act as an mRNA translator to assemble proteins from amino acids. So imagine you have a strand of RNA, and it comes in and it comes out as a protein. It comes out as a protein. It comes in as a strand of RNA. But more about protein synthesis in Unit 2, of course. Centrioles are small tubes of protein fibers. There are a pair of them next to the nucleus, so they take part in mitosis, and they move the chromosomes apart. I'll talk about more of them in the mitosis tutorial. In terms of what organelles you need to remember definitely the nucleus, definitely the endoplasmic reticulum, definitely the Golgi Apparatus, and the chloroplasts and lysosomes. It's probably best to learn them all, because, well, all of them are listed in the book, so it's common sense to learn all of those basic molecules. So if we go back to our pictures now and have a look at the centrioles, just these small bundles of protein fibers like that, and the microtubules is each fiber, and they flex around in order to keep the cell fluid. So that's it for this tutorial. If you stick around, there'll be another tutorial about how organelles work together and the idea of division of labour. So hope you found that useful, and thank you for watching.